Thank you very much for the kind invitation and to be part of this great meeting and this session. I'll take you now to yet another part of the brain. We've been at the front and we've been down in the middle, and we're now going right to the back. And I will give you some data which might make you think for decision making. A lot of it, if not all, is happening there. My lab is investigating the neural basis of perception and decision making, and in that we are probing a very fundamental process of the nervous system to receive, combine, and rearrange incoming information to support behavior. And the perceptual system, in particular vision, is a really good test bed for these decision making questions, because we can deliver very precise external stimuli with a precisely defined content. And in doing so, we can study the principles underlying these fundamental operations. And we do this in my lab across primate species, so we can go from neurons to behavior. All the three experiments I'm going to talk to today involve monkeys or humans making decisions about such bistable figures as you see on the screen right here. Which is basically a rotating cylinder, and you may see the front surface left or going to the right, and it should switch occasionally. We can disambiguate this stimulus very easily by introducing binocular depth. So we're basically moving the left and the right, but moving dots into different depth planes. So for instance, here, if you move the leftward moving dots in the near surface and the rightward moving dots in the far surface, we get a clockwise rotating cylinder as viewed from above. And both monkeys and humans can discriminate this conjunction between two sensory cues, this depth and motion very, very well. Very small amounts of binocular disparity can disambiguate this. This is a curve, a psychometric curve from one of the monkeys in my lab, but the thresholds between monkeys and humans in these tasks are comparable. In the three experiments, I'm going to look at three types of bias induced in this perceptual task. First, a manipulation using direct electrical microstimulation in the brain, manipulating, biasing the percept in a particular direction. And then I will use this access site in visual cortex to show you that reward bias, of which we've seen a great example yesterday in Long Ding's beautiful talk, which is very powerful across primates, that some of this is mediated in biasing signals already in visual cortex. And in the last part of the talk, we are turning to developmental studies in kids, looking at social influence and how it biases perceptual decisions. And the take-home message from all this will be that a lot of this bias that we see in the behavior is already influencing the filtering and the integration of sensory information back in visual cortex. So we're starting in area V5MT, which is this red area, highly myelinated area on the scan from one of our monkeys, buried in the superior temporal sulcus in the monkey here. That's the front and the back, for those of you not so familiar with the macaque. When we record in this area, most of the cells are superbly tuned to direction of motion in a particular part, the receptor field in the contralateral visual hemisphere. And most of them are also tuned very well to binocular depth. Here, a case of a neuron tuned to near depth. And these is individual cells, which we got by ice fishing, as Pierre nicely put it, are actually organized in columns. So we have isodirection columns and isodisparity overlaid of these clusters. And this is quite important to actually make the microstimulation experiment work. So if we look from the top onto the monkey in a cartoon view, and here the monkey is fixating on the screen, you can imagine a receptor field, in this case a multi-unit one, because we are in a column, as in a specified area of the contralateral visual field at a certain depth, in this case in the near plane, and sensitive to a certain direction of motion. And with that, it would here respond, if viewed from above, to a clockwise rotating cylinder, but not to a counterclockwise, even though the motion and depth content is the same on the screen. It's just displayed in a different conjunction of motion and depth. Now, in the task, in the initial task here, we have the monkey fixating on the screen. Cylinder stimulus comes up in the multi-unit receptive field. It's matched to the location and the sensitivity of this multi-unit selective field. At the end of the trial, the monkey indicates the direction in which the cylinder rotated by making an eye movement to one of two choice targets. Here, for example, to the left for a cylinder rotating, the front surface rotating to the left. The animal gets rewarded for correct choices, correct in terms of the visual stimulus only. On half the trials, we electrically microstimulate a single site in V5MT inside a column of same tuning, probably stimulating with a small current of 20 microamps, 200 hertz, about 5 or 10 neurons. Key thing is here, we reward only for correct choices to the visual stimulus, not the microstim. So we give the monkey every incentive to ignore the microstimulation if it's, for instance, a weird signal. So here you have the behavior results, part of the behavioral results for one stimulation site. That's a single experiment. Percent choices counterclockwise on the y-axis and a different cylinder stimuli we used on the x-axis. These are only the non-stimulated trials, and you can see here that the monkey can discriminate quite well counterclockwise from clockwise. On half the interleave trials, we stimulated a site with a preference for counterclockwise rotation. And now for these trials, you can see we have been able to bias the perceptual choices consistently in the counterclockwise direction. And not just for the bistable stimulus I showed you in the very first movie, but even for stimuli that were clearly unambiguous when non-stimulated. And the behavior is as if we had added more visual sensory information for a particular percept. And just to show you that this has nothing to do with the behavioral response or just boosting a visual signal in the near surface, here are two stimulation sites with preference for rightward motion, but one rightward motion in the near surface and one rightward motion in the far surface. And here you can see the behavioral bias with microstimulation is in opposite directions. So indicating the opposite percept and making an eye movement response in the opposite direction. So we are clearly on the perceptual sensory side affecting the appearance of the stimulus. So in this body of work, together with a lot of neurophysiology, we have characterized the site in V5MT in an extrastriate visual area that represents the perceptual evidence for this task. And this is obviously in the sensory motor loop for perceptual decision-making for stereo and motion. That's been well characterized by us and others from sensory information to the decision variable to the decision response. And we can use now this point where we have the specific access point to ask questions about other forms of bias that affect perceptual decision-making. And we're now going to a reward that's been mentioned in quite a lot of the talk and is quite widely studied, and particularly the effects of reward on making different choices. So if we, in a very simplified way, look at the arc of this very simple decision-making process from sensory information to behavior, there's quite a lot of evidence that reward signals are present in neuronal and MRI signals, where integration of sensory information is happening where the decision-making variable is computed in area LIP, lateral interparietal area, and parietal cortex, and prefrontal areas. What we were interested in whether some of these signal comes from reward integration earlier in the pathway. At the level of perceptual evidence. And we use the microstimulation in this experiment here to dissect the pathway into halves, before and after. So the idea is if reward acts before this stage where we put the artificial electrical signal in, it might be able to differentially up and down regulate the sensory information. And we could see a dissociation between the size of the microstimulation effect and the size of the reward signal. However, if the reward only acts later, beyond where we microstimulate in higher visual areas, then spikes are spikes, the artificial and the inserted and the sensory encoded ones, and the microstimulation effect should not be changed. To up the ante again, we are not using a reward schedule that rewards differentially left and right responses, but using a pattern to which the monkeys are very familiar of giving higher rewards to consecutively correct responses. So here's an example. So if the monkey gets 0, 1, or 2 choices in a row correct, the reward is low. But if he gets 3 or more correct, the reward goes high, much higher. And the monkeys are familiar with this through training over tens of thousands of trials. So there's a predictable pattern to high and low rewards across the different trials. By itself, without microstimulation, this doesn't induce a bias. The only thing we see if we average for one monkey here over thousands of trials is that maybe the curve is a little bit steeper for the large reward trials. So now we are back to the experimental task where the monkey made a choice about the cylinder within eye movement. On half the trials, we electrically microstimulate in V5MT an identified column, and we reward for correct choices, only correct with the visual stimulus. I should add for ambiguous trials, we rewarded half the trials at random. So here you have the data I've shown you before, with the non-microstimulated and the microstimulated trials. And now we separate the trials out for this individual experiment at this one site to trials where the monkey was receiving, expecting at the end of the reward a low reward or a high reward. And you can see immediately for the single site that when the monkey expected a small reward size, on these trials, the microstimulation effect is much larger than on those where a large reward was available at the end of the trial. So it is as if when the monkey expected a large reward, the sensory incoming sensory information was stronger than the fixed microstimulation effect we had on these trials. And to quantify this intuition, we fitted to the behavioral data a simplified drift diffusion model. You saw quite a lot of them yesterday, but unfortunately not in this session. So just as a brief reminder, information is integrated over time for the two decisions, in our case, counterclockwise or clockwise rotation, until the information reaches a bound which unleashes the decision. We use here and in the next experiment, particularly two variables, the variable K, which is basically sensitivity scaling the incoming sensory evidence is related to the drift rate, seen to be on the sensory side, and the decision to bound, which gives you the distance, the time over which the information is integrated, and is generally seen as a parameter that might be neuronally implemented, for instance, in priors in parietal cortex. So if you fit our behavioral data, both changes in K and B in drift rate or distance to bound change the slope. But what if we try to fit our microstim paired data? And here it's very clear. Changes in distance to bound can only change the slope, not the size of the change of the microstimulation effect. But changes in drift rate, or factor K, affect the size of the microstimulation effect. And when we want to fit the combined pool data from our two monkeys here on these microstimulation reward interaction effects, we need, in order to explain the data, both changes in distance to bound with reward, expected reward, and changes in K to distance, changes in K and drift rate in order to fit the data. So, so we need an effect on sensory evidence, and there's clearly the impact of the artificially induced neural signals is affected by expected reward, and we can model this in a way that suggests we modulate the weight of the incoming visual information, so that the reward effects are already seen in these earlier visual signals. And basically what's happening is it's not just the behavior that is nudged, that I would choose the higher rewarded condition choice when I'm uncertain or so on, but I'm already filtering and shifting the weights on the incoming sensory information. So now to the last part, looking at social influence on perceptual decision, and we switch now to a human experiment. So social influence is a very powerful cue, and it's just a brief reminder on a couple of experiments here. So Solomon Asch has shown that people very readily change their perceptual choices, very simple perceptual matches, to an incorrect, clearly incorrect majority. And we know from studies by Subiol and Terrace that monkeys can learn tasks by watching others. So here we again are with a cylinder task. This time we studied a cohort of 155 children aged 6 to 14, and they carried out a cylinder task and a reaction time task on a touch screen to make it easier for the youngest kids, touching left when the front surface goes left and right. There was also a backstory with black holes and being a spaceship pilot. But before the cylinder was shown, there was a short video clip by gender-matched advisors who told the subjects whether they thought the cylinder or the black hole was rotating left or right. And here is one of the pooled psychometric data from a cohort of about 30 kids, 12 to 14 years, plotting the proportion choices left again over the different cylinder disparities. This is now normalized cylinder disparity to account for different thresholds across subjects. And you can see that the advice for left influences the judgment across all the range of disparities to making the kids making more choices left, or the teenagers here, and more choices right. So there's a very, very strong systematic integration of social advice, social influence in this perceptual decision-making process. If we look across the age groups from 6 to 8, 9 to 11 to 12 to 14-year-olds, it is very, very striking that it's particularly in the teenagers you find a systematic integration of social advice, and not in the younger age groups at all. And that is, we also see that whether the advisor is a gender-matched adult or if it's a peer of the same age, where we have the systematic integration for the teenage years, but not earlier. And that coincides with the age in the teenage years, where other groups have shown with imaging an increase in white matter connectivity from prefrontal cortex back to parietal and visual areas. We also had an IQ and age-matched autistic cohort where we don't see this systematic integration in the teenage years. We rather see across all the ages a small bias towards the social advice. But here again, imaging studies have shown that this increased connectivity with prefrontal cortex and also the volume increase in prefrontal areas that might be relevant here for this top-down control is less than in age-matched typical children. Again, we fitted the behavioral data with a drift diffusion model and looked at the different impact of the drift rate, which is supposed to affect sensory processing, and distance to bound, which is thought to influence a starting point. And what is quite clear, for all these models, we need drift rate and distance to bound to vary with social advice. But we get significant effects in the neurotypical children, teenagers, 12 to 14, explaining the effects we see with social advice on drift rate in these groups only, not in the other groups, and not the autistic children. And the small bias we see at other ages is best fitted by changes in distance to bound with social advice. So again, this points to that we need to change the weighting on the sensory information, which at least partially contributes to the bias we see in those children with social influence in their decision behavior. So overall, I've taken a well-studied circuit in the monkeys looking at perceptual decisions where we can manipulate in the animals very precisely the perceptual appearance of the cylinder by direct intervention in the brain. And linking these sites and these approaches through common behavior and common neurocomputational models, I've shown you evidence that expected payoff for the opinion of others might actually affect visual processing, and it might do so by changing the weights on the sensory signals by the expense, on some sensory signals at the expense of other sensory signals. And so the decision-making biases we see in behavior might be at least partially mediated by changes in perception. And these processes have relevance to clinical conditions like autism. And so the key thing is now to going out to the circuits and integrate this with top-down signals from areas, frontal and parietal, back into visual cortex that can link these general motivational signals in the specific structure that shape perception, the particular decision. And generally, this is a well-controlled framework with a very simple task, but allows us to address these questions from the single neurons to circuit to behavior. Oh, and my last slide, not the least, this is mostly the work of Nela Sismal and Imogen Large, who were grad students and postdoc in my lab in Oxford, in collaboration with labs in Australia, Germany, UK, and the US. Thank you.
Thank you very much, Kristine, for this outstanding cross-species talk. Can you hear me okay?
Yeah, I can hear you.
So let me shoot the first question. It's about the difference between an effect on perception versus an effect on decisions. So I get the argument from drift rate versus distance to bound when you feed the DDM. But in monkeys, obviously it's not possible, but in humans, would it be possible just to ask participants whether their perception has changed or whether they just adapted their response to match the advice?
Yeah, it's a very, very good question. I'm not that much for subjective experience, but it's been done back to Solomon Asch, and people maintained, even with these lines, that people had seen it differently, not everybody. And we did ask the kids, and I mean, the instruction was very clear, to report as accurately as possible. And we have also done rewards choice. We didn't have time to talk to about today, reward in humans. And we instructed them, and they got monetary reward, and only for accurate responses, and they got feedback. And they still report the way they do, which makes all this taken together, does point to that they're seeing it differently. And we didn't have any report otherwise, but we haven't systematically interrogated them. The problem is if you do it at each choice, you get them to get very suspicious of you. That's the other disadvantage of humans. They're very suspicious. But there is plenty of evidence from this that they report what they see, because otherwise they lose rewards. And the monkeys actually lose rewards when they go with a microstim, or when we had another study, unequal rewards, and they lose rewards if they're inaccurate.
Thank you. So we have questions from Lawrence Hunt. I wonder whether reward priors in MT are expressed only during choice. And second part is, was microstim always delivered mid-decision, or is there evidence for any similar effects on choice if delivered pre-decision?
Oh, this is a very good question, of course, from Lawrence. So we have no evidence in the neuronal signal prior to the onset of the stimulus of any changes. And not for the want of looking so far, but we need to look at this again. There are definitely effects in the early trial of outcome on the previous trial. But this is not the case in the data I've shown you here. So there is early on in the trial a signal increase for previously preferred rewarded signals, which is counter to adaptation. But it's not there prior to the onset of the stimulus, which again points to a gain change. And then the second part, we should actually, that's something we are doing and are planning to do in experiments here in Magdeburg, delivering the microstim at different parts of the trial. We haven't done that. There are other studies which show it's the most effective during stimulus presentation for the integration. And you can't do it yet by microstim alone. It's also something we're working on. But so when people looked at, it is during stimulation, it's the most effective in these visual areas.
Okay, thanks. Another question from Longding. Interesting that adult advisors had the largest effect. Does it reflect the perceived reliability of the advice?
Yeah, that's a great question. We didn't ask our kids on the reliability, but there are other studies from people, for instance, in Göttingen, which say that that has an effect. We can't really distinguish the size of the effect very readily, and it was 2 different cohorts, so this might be the case. What is also quite interesting, I didn't have time to talk about, the youngest kids for the bistable stimulus went counter to advice, and particularly when it was the parents, which I think an adult, which would no parent surprise, I think. It's something other studies have shown, yes, the reliability makes a difference, but we didn't look at it in this study. However, teenagers, actually peers are the stronger effect. That's work by Sarah Jane Blakemore in London.
Right. Last question maybe by Johannes Argemisen. Was the reward stake high versus low predictable across trials or randomly fluctuating? And did you see reward inducing some bias in choices, left versus right?
Okay. So it was the high versus low was predictable across the trials from these patterns, and we used within a day at least the same reward level, and it was pretty much the same between monkeys, and they were quite experienced at this. So the pattern where when they did a task and when asked to respond, the pattern of so and so many correct or incorrect to go high, it was well entrained. And they were doing this in a block. Did you see the reward inducing some bias? Not in this experiment, because they were obviously, it depended, the choices direction did vary. But we did a separate experiment in monkeys. We have behavior from a couple of them, and you can induce the same kind of difference in reward size when you give different rewards for left and rights and block, as Longding has shown yesterday, and we have data on our own. And that's obviously the next one, looking at whether these signals we see early on in this paradigm, whether we can see them at the MT level for the different choices. And we have some pilots that, yes, we do.
Right. So I think it's time to have a general discussion. So I don't know if the other speakers can